So, good evening. In today's session, we will be talking about the week 6 topics, and we will also be practicing few MCQs related to week 6. So, in week 6, you have looked into the what is automated data processing, you have looked into workflow of automated data processing, you have also hands on the firecloud and what is the advantage of firecloud, what you can do with firecloud, and what are data models. And you have also looked a little into the big data, that what is big data and how to handle big data and how it is formed. So we will, along with this, we will also look into the current literature, and I'll take your doubts regarding the current assignment. So let's proceed. So the first topic is data processing. So before moving forward to automatic data processing, we will firstly look into the basic concept that what is generally data processing. So when we talk about data processing, it is a series of operations which are performed on data to transform, analyze and organize it. So when you see a data, it is a raw form, it's not organized. So when we try to organize it and try to take out the useful information out of a data, then it is known as processing. So data processing is like when we perform certain operations on our data to transform it, analyze it and organize it. So there are different stages. So one such stage is collection, then we'll filter the data, sorting and analysis. So now let's look into these figures. So when we say what is data processing, so it is a collecting and manipulating of data into usable and desired form, so that we can understand that what is there in the data. So that's why we have to perform processing on that data. And if we look into the processes which are involved in the processing, so firstly we need the data. So we collect the data, then we prepare the data, because in your data there could be multiple things such as noise, contaminations, so you have to remove those things. So that is known as data preparation, when you remove the noise and you kind of pre-process it, you prepare it so that further analysis could be performed on that data. Then comes data input. So after preparing your data, you will input your data to some processor where you will be processing it. Then comes the data processing step, where you will decide that how you want to process that data. So there will be multiple conditions depending on you. You will design some workflow how to process the data, and after processing the data you will get some output. So now the next step is to interpret that output. So after obtaining the output, you will interpret it, that what you can analyze from that information, and ultimately you can store this data for further use, or you can share this data. So these all are the stages which are involved in data processing. Now let's look into the data processing types. So automatic data processing is one of the subtype of data processing. It depends that what you are going to do with the data and how the data is being processed. So on the basis of that, there could be multiple types of data processing. So another such type is cloud computing. So cloud computing is also one kind of data processing when you are doing it on the multiple computers on the internet which are connected via internet. So that is known as cloud computing. So these all things, these all types are somehow linked to one another. These are not like you cannot do automatic data processing in cloud computing. So these all are linked. It depends that what you are doing in the processing. So that's why the name is given on the basis of that. So automatic data processing is the data processing where you have automated the process, you are not doing it manually. So you have designed some workflow and you are letting it done automatically. Then there is batch processing. So batch processing is like you are not dealing with the entire data in the input, but you are just dividing it into small batches. So then you are dealing with batches. So batches is a small set from a bigger set. Then comes real time processing. So it means that when you are processing a data in the real time. So in certain cases you can assign the tasks and your data can be processed at some particular period of time, but when you are doing it on the real time, so that is known as real time processing. For example, at the same time you are collecting some information from your machines. For example, you are running your spectrometer and at the same time you are collecting the information, so that is known as real time processing, that information is collected at the same time. Then multi-processing or parallel processing. So when multiple processes are being run at the same time, for example, you have a same data but you have defined multiple processes, that in one process you are capturing one feature, in another process you are capturing another feature, or some other process you are doing on it. So when you are running multiple processes at the same time, that is known as multiprocessing. Similarly, online processing, if you are performing it online, then manual data processing. So this is done manually. For example, you have an Excel sheet and you are performing certain things manually, that you are calculating everything and then you are giving it for the further step. Then mechanical data processing, similar to manual processing only. Electronic data processing, in which uses some electronic devices to perform the processing. Distributed processing, when you are distributing it across devices. So these are some types of data processing techniques or data processing types, you can say. So now we'll discuss about automated data processing. So as the name suggests, automated, so it means that in this, the data which you provide, this will be automatically collected, organized, analyzed and transformed. That means each processing step will be automatically done on that data. So it involves the use of algorithms, scripts and systems. So you have to design certain algorithms. In algorithms are kind of steps. For example, you want to perform a task, so there will be multiple steps which are required to perform that task. So algorithm is like a program which will tell you stepwise things to do. So for automated data processing, you have to design algorithms or you have to write scripts, and then you require a system where you want to run those scripts, so that you can streamline the tasks. You can decide that what will be your data entry, how the cleaning will be done, how the analysis will be done. So in this way, automated data processing is done. So why it is used? So it increases the efficiency, because in manual thing, when some person is doing some data processing, so there are chances that there could be some errors or some mistakes could be done. But in case of automated data processing, because you are writing scripts, so if your script is right, then your data will be accurate. There are lesser chances that there will be some errors. But it depends on the script. So if there is mistake in the script, then it can disturb all the processing. But if your script is entirely correct, so it will not do any errors, so the data will be accurate. And then you can also obtain the speed, because manually entering everything is like time consuming. But in automated data processing, as each step is automatically done, so it's quite quick. So that's why it will provide you speed in handling large volumes of data. So that's why we choose automated data processing over the manual data processing. So here is an example that when you should choose automated data processing. So for example, you have a large data sets which you cannot do manually, so for that time you can choose automated data processing. Then if you have repetitive tasks, so for that also you can choose. For example, you have a real time data, so real time data also can be processed with the help of automated data processing. And it is also cost efficient along with scale. That means if you have a large amount of data, then also it won't cost you much. Then data integration requirements, so if you want to integrate data from multiple sources, then automated data entry is better. If you require accuracy, predictable and structured data, and then compliance and audit trails, long term data management, so these all are the things when automated data processing could be opted rather than manual data entry. So is it clear? Okay. Now here are the comparison between automated data entry and manual data entry. So in terms of accuracy, as I've told that you are writing scripts, so scripts will perform each and every task with the similar way, with similar speed, similar efficiency, similar accuracy. So in that way it is highly accurate. But in manual data entry, for example, a person is entering the data, so if data is huge, then at some point of time that person might get tired, and there are chances that he could incorporate some errors, so it is prone to errors. If we talk about speed, so automated data entry is very fast, whereas in manual it might take hours depending on the size of your data. Then about cost, so automated data entry is quite cheap, whereas manual data entry is expensive, because if you are hiring a person and asking him to perform certain tasks, so he will also charge you some means wages, or you have to provide him some salary, so in that case it is quite costly. If we talk about scalability, so it's difficult to train multiple people, because if you have a large amount of data and one person can't handle it, so you have to train multiple people. So in that case, scalability is quite difficult in manual data entry, whereas if we talk about automated data entry, so in that case you can easily scale your data set and it will easily handle it. Given that you should have those machines and processors to perform that data processing. Then if we talk about flexibility, so in case of manual data entry, it requires training. It's quite flexible, but for example, in automated data entry, if you want to change something in your, means for example, now you have decided that I should choose this threshold for my selection of the mass to charge ratios. So in that case, you require entire reprogramming. You have to reprogram the script which you have written. But in case of manual data entry, you simply require training a person and asking him that make sure this should be the new criteria. So in that case, it's little complicated in terms of automated data entry because you have to reprogram. So in that case, it's not that flexible, because in certain criteria you might change multiple things of your that algorithm. So in that case, reprogramming becomes a bit difficult. Then if we talk about regulations, so in automated data entry there are no laws which are tackled, but in case of manual data entry there are some labor laws. You can't ask a person to sit for entire day and perform your work, whereas no such laws are required in case of automated data entry. Then integration, so for example, you want to integrate multiple softwares, or you want to integrate different data from different repositories, so that is easy with automated data entry, whereas in terms of manual data entry it requires extra person to do that. Then if we talk about organizational change, for example, if organization is changed, so in that case automated data entry will not be affected, whereas manual data entry will get affected because now institute has changed, organization has changed. So in that case you have to means arrange for that changes. So that's why if we compare overall, so automated data entry is better than the manual data entry. Now let's talk about cloud computing, which is also a type of data processing. So what is cloud computing and why it's given the name cloud? So as we know that cloud is on the top, which means that in this case there will be multiple devices which will be connected to a single big cloud. And means cloud is not a original cloud, but it means that multiple devices are connected wirelessly, that's why it's termed cloud. So for example, there are some servers, virtual desktops, software platforms, applications and storage data. So all those things are available at a single place, and it is coming from multiple desktops which could be present all over the world. So from different places, when those devices are connected through internet and they are sharing their data, they are sharing machines, virtual machines, so all those things comes under cloud computing. So it is like on demand delivery of computing services such as servers, storage databases, networking, software and analytics over the internet on a pay as you go basis. So yeah, it will cost you to some extent depending on that how much space you require or how much processors you require. So what happens that bigger companies, or there are some organizations which have high functional processors, high functional devices, so they do not require that much space. So in that cases what they do is they kind of provide that space to the people. They allow them that you can assign your task to those processors or those machines, and you can rent those processors or storage spaces for some time or depending on your need. So in that case you can demand, and they will provide you with the computing services which can be in the form of servers or storage. So as we all know that nowadays we use Google Drives or Google Photos to store our photos. So in that way these all comes under cloud computing, where you are storing things in a one place and you are paying for it. So some space is free, but after that you have to pay if you want some more space. Similarly in terms of other processors or bigger machines, so in that case you have to pay depending on for how much time you want to perform that process. So in that way space will be allocated. Then cloud based storage makes it possible to save files to a remote source. So why it is called remote source? Because even if you move from one place to another, then also you can access it. So it's not local. When we say local source, it means that it is on your device, which for example I am working in a organization, so where my data is stored in my local PC. So when I go to some other place, so I can't access that data because it is lying on my local machines. But what help that cloud computing provides you, that in this case it stores the data at the remote source. So even if I move to any place in the world or anywhere in the world, I can access my data from any place. So that is known as remote source, which can be accessed remotely from any place. It is not fixed. And cloud data storage platform includes Google Drive, Dropbox, OneDrive, and Box. So these are some of the examples. Later on we'll see more examples of this, that which are the cloud computing service providers. So yeah, here are few examples which you might have seen in your week 6. So first is Google Cloud Platform, then Amazon Web Services, Microsoft Azure, Oracle Cloud. So there are many, but I'll be focusing on mainly 3 of these service providers. So first one is the Google Cloud Platform. So what are the different advantages it provides you? So it helps you with compute services. If you want to perform analysis on big data, you want to store big data, so all those things can be done on Google Cloud. Then Cloud AI, which is the artificial intelligence based processing. Then storage services, you can store your data. Management tool, if you want to manage your data, you want to organize it. Then Internet of Things, so it's like there will be multiple things which are linked to one another. Then networking, so if you want to connect multiple devices on Google Cloud, that also you can do. Security and identity management, so it's quite secure, so in that way you can use Google Cloud for these purposes. That means your data is safe. If you are storing something, so nobody can steal it. Then comes Microsoft Azure, so it is provided by Microsoft. In this, security is given, word access, so you can access the data from anywhere from the word. Scalability, so you can perform processing on the larger amount of data or bigger data, so it's scalable. Then hybridity, flexibility, so it's flexible. These all are the features of Microsoft Azure. Then comes Amazon Web Services, so similarly in this also you can get the servers. So for example, you want to perform multiple, you want to host a website, or you want to perform multiple bigger tasks, so you can ask for the servers. Then networking, storage, remote computing, mobile, email, security, mobile development, so all those are services which are provided by Amazon. There are multiple services as well, so these are few of the services which are offered by them, and main thing is that in all of them they ensure your security of your data, which means nobody can steal your data or nobody can harm your data, nobody will be able to access your data. So it depends on you. You can there is a feature which helps you, that if you want to private your data, so nobody could see it, but in case of data sharing, in sharing also there are some guidelines. So in that case you can mention that how much access you want to provide to someone, and for example if you want to provide access to a limited number of people, so in that case also you can ask them to generate some passwords, so that anyone with the password can only access the data, other will not. So security is one of the important feature, because that is the main concern while storing the data on cloud. So is it clear till now? Okay, thank you. Let's move. Okay, so now let's see automated data processing workflow. So what is a workflow? So workflow refers to series of sequential steps or tasks that are defined, automated and executed to achieve a specific outcome, such as collecting, cleaning, transforming, analyzing and reporting the data. So as the name suggests, workflow, that how work will be done in a flow wise manner, so in a sequential manner. So it will tell you about the sequential steps, and each step in the workflow, it represents a logical phase of the process. So logical phase means it will tell you each and every phase, that means what is input, what are the processes and what is output. So there are 3 main phases in data processing. So workflow will tell you all the 3 phases that are managed and automated to streamline your data processing and ensure consistency, efficiency and accuracy. So here are listed some of the steps which are involved in automated data processing. So first step is data ingestion, that means you have to collect and import data from various sources. So in terms of automated data processing, you do not have to manually enter data or collect data, you can simply write algorithm. For example, if I want to analyze the cancer specific data, so I will simply write an algorithm in which my program will itself collect the data from multiple sources, such as it can collect it from TCG or other repositories which are already available. So first step is data ingestion, in which we are collecting the data or importing the data from various sources which could either be databases. For example, in my local computer only if I have data, so then also I can write the code, or from APIs or other files into a centralized system. So there will be some system which is performing the processing, so that will to that particular folder or to that particular site my data will be imported. Second step is data validation and cleaning. So now this will automatically check the data quality. It will remove any duplicates if there are some duplicates present, or if there are missing values, or if there are some kind of contaminants or errors are present, so that will be collected in my second step. They will be removed, they will be corrected. So now my data is prepared. Next step is data transformation. So data transformation is a step which will convert our raw data into a structural format, which could involve filtering, aggregating and normalizing the data. So for example, when I am collecting data from multiple sources, so there are chances that my data is not in a proper manner, in an ordered or structured manner. It may vary across different sources. So in such conditions we need to normalize our data so that it could be in a normalized or uniform manner across all those from where their source is. So for that we require data transformation, but if I am collecting data from a single source, then this step is not needed. So it depends that how your data is and which step you want to perform. Then fourth step is data integration. So this will combine the process data from different sources into a unified data set or data warehouse. So similarly, if I want to integrate data from multiple databases, then I will perform data integration. So these all steps are mentioned because these all are the steps which I can use in terms of big data processing. So means these are not the necessary steps. I will not always perform them, but it depends that whether I need them or not. So data integration, so if I want to integrate data from multiple workspaces, so in that case I require data integration step. Fifth step is data analysis and modeling. So now I have obtained all my data, it is already transformed. Now I will perform analysis on it. So for analysis we can use statistical methods, machine learning models. So in the previous session we have seen that there are different statistical methods which can either tell you that whether there is a proper difference between two data sets. For example, if I am comparing the normal and the cancer patient, I want to see genes which are only expressed in the disease state. So in such scenarios I can compare, I can use statistical methods to prove my alternate hypothesis that what I am hypothesizing is correct. So that is also one of the step under data analysis, where you are applying statistical methods to check that whether what you are expecting, whether it is true or not. In terms of machine learning models, if I have a data set and I want to predict certain things, or I want to classify my data, so I can use machine learning approaches. So last time we have seen supervised and unsupervised learning methods and different techniques which they involve, or other techniques to extract the insights from the data. So this is about my fifth step, which is data analysis and modeling. So now my data is being analyzed. So my sixth step will be results generation and reporting. So now after analysis I want to see my results. So results can be in the form of tables, or it could be in the form of graphs, or it could be some reports. So in that way results will be generated. And it's insights. So for insights means I can observe those graphs and I can generate insights. So results can be analyzed by ourselves, or for example if we have certain criteria that if p value is higher than or p value is lower than this, then I will accept. So all those things comes under the interpretations. So that can also be means combined in the workflow of the automated data processing. Then comes automation and scheduling. So these all steps can also be automated. For example, if I want to run same analysis again and again, so I want to kind of repeat my task again and again, so in that case I need to automate it and schedule it. For example, I am performing analysis on TCGA data, and after some time my data gets updated. So in TCGA maybe some new genes come up, so I want to again perform the similar processes to make sure that I am also updated. So in that case I can schedule that after this particular time again reprocess it, again do the processing, again perform all the steps and automate my data. So in such cases we can automate and schedule that after what time you want to perform the processing. So all this comes under automation and scheduling. Then comes data storage and archiving. So data storage means after all the analysis have been performed, so I have to mention that where I want to store my data. So this data storage comes, for example, if I am performing my automated data processing on a cloud computing. So in that case, I can make sure that where I want to store my data, whether I want to keep it in the remote device only or whether I want it to get downloaded to my device. So in that case, I can uh, means mention that where I want to store the data, or whether I can also create means another database in my cloud computing only to store my data. So in that way, and archiving means I can archive it and later on I can de-archive it and access my data so that I can store it in a smaller place. Then comes feedback and optimization. So after performing all the processing, if my results are not that accurate, so in that case I can provide feedback and I will try to optimize my algorithm so that it could perform better analysis. So I can enhance the performance by feedback and optimization. So there are some machine learning models which learn after each and every step, so they try to optimize model each time. So in such scenarios, when I am using machine learning algorithms, so I can simply feedback and optimize my analysis. So in this case, it will continuously monitor the workflow performance and adjust the processes to improve the efficiency and accuracy. So this all comes under automated data processing workflow, that what are the major steps which need to be performed. Then there is another term which is pipeline. So many people get confused that what is a workflow and what is a pipeline. So they are nearly similar. Workflow is a bigger term, pipeline is a smaller term which comes under workflow only. So pipeline, when we say pipeline, so it means that these are also the series of sequential steps or stages that are automatically process the data from an input to output. So when I am completely so when automatically my workflow is processing everything, so that is known as pipeline. It will begin from input and then it will give me output. So it might incorporate multiple workflows in it. So pipeline is like complete automated thing, whereas workflow, it might not be automated. So workflow is simply a sequential representation, but pipeline is like automated workflow. So this is a smaller section under workflow where everything is automatically processed from input to the output, and each stage in the pipeline performs specific operations, and output of one stage is the input to the next. So there will be multiple stages. So now we will see this particular example, then you will be more clear that what pipeline means. But pipeline is like a bigger thing in which entire thing is automatically connected to one another. So there will be multiple stages, so each and every stage will perform some kind of processing. So when one process is completed, so its output will be acting as the input for my second process. So similarly there could be multiple processes which are running one after other, so they will be sequentially running. So that is known as pipeline. So unlike journal workflow that may include manual steps and decision points, so in workflow you might include certain decision points, but in case of pipeline it is like completely automated with each and every step designed to perform a specific function. So these are two stages of a single pipeline. So now we will look, these are obtained from one of the papers. So now I will explain them in the next slide. So there was a paper published in Proteomes which is about quantitative proteogenomic characterization of inflamed murine colon tissue using an integrated discovery, verification and validation proteogenomic workflow. So in this paper they have looked into the chronic inflammation in the colon which can trigger expression of non-canonical protein sequences which contribute to cancer development. So in the previous assignment you looked into the non-canonical, that how you can identify the non-canonical protein sequences when you do not have a database, because in mass spec you require a database which already contain all the protein sequences. But what if you have the non-canonical protein sequences in the dataset? So how you can perform the peptide matching when you do not have sequences already available in the databases? So similar thing they are also doing here. They are trying to identify non-canonical protein sequences which are contributing to cancer. So in this study their proteogenomic approach used the customized FASTA database generation. So they have a transcriptomic data which is the data at the level of RNA. So now they are customized generating the FASTA database. So they are converting that transcriptomic data into a proteomic data so that non-canonical peptides can be identified. So in the bioinformatics workflow, they have identified 235 putative peptides, out of which 58 were verified and 39 were validated by targeted assays, which highlight that integrated approach for identifying non-canonical peptides via Galaxy platform. So in the week 6, you have also looked into the Galaxy platform. So it is also kind of a graphical interface which performs multiple processes. In this, you do not have to write code separately. It is like a bigger platform in which multiple analysis you can perform on your data set. You have simply have to enter your data and follow the instructions which are given in the Galaxy platform. So similarly, these things they also performed, and they identified the non-canonical peptides. So now we'll see that how they have done. So if you look at the outline of their experiment, what they have done in journal. So they have two mice, one is control mice and another mice was subjected to 5 months of H. hepaticus infection. So in infection, it leads to colon cancer, and they want to identify the peptides which are observed in case of cancer. So in one mouse, they infected it with H. hepaticus. Now it will lead to inflamed proximal colon tissues. So from this also they will obtain mRNA, and from normal proximal tissue also they will obtain some mRNA, so that they can distinguish between the control and the cancer state. So now protein was also isolated. So this is a proteogenomics approach. That means they have collected protein also and mRNA also, and they are trying to integrate it. So for the protein which was isolated, they will perform trypsin digestion, and TMT 6-plex labeling was done to label them separately. Different proteins were labeled separately, and then LC-MS analysis was performed. So this comes under the proteomic analysis. Whereas similarly, when they obtained mRNA, so they have sequenced it, RNA sequencing has been done, and it has been converted to the customized protein databases, because we can look at the codon table and construct the protein sequence which could come from the mRNA sequence. So similarly, they have customized protein sequence databases have been generated. This is just to identify the known canonical proteins which are not present in the database. So for that purposes they have done this. Now let's look at the first stage of their pipeline. So first workflow. First step is generation of RNA-seq based custom protein FASTA database. So as I have previously mentioned, that their aim is to identify known canonical peptides. So if they are completely relying on the database after MS, means after obtaining peptides from MS, if they rely on the already available database, so in that case they might not be able to find the non-canonical peptides which are not present there. So for that they want to look for all possible peptides that could be generated. So for that they are generating this custom protein FASTA database. So first of all, there are FASTQ files which are present in forward and reverse. So these FASTQ files are the FASTA quality files. They are generated after performing sequencing of that RNA-seq data. So after sequencing, it is checked that whether the sequencing is done correctly. So after that, the end product is FASTQ files, which are the quality files, which tells you that sequencing results are correct. So now forward and reverse sequences will be obtained from the RNA-seq data. Then comes HiSat2. So what is the purpose of HiSat2? It is a fast RNA-seq read alignment tool that maps the reads to the reference genome and integrates with freeways for the variant detection. So this is a read alignment tool. You already have a reference genome. So in that genome, you try to align it. So after obtaining the sequence, when you are aligning it to a reference genome, so that is done by the HiSat2, which is RNA-seq analysis and assembly. Similarly, I have mouse genome GTF file. So GTF is a format which tells you about the gene information. So it keeps information of entire genome. Then comes mouse proteome. So in SwissProt, because they are performing analysis on the mouse, so it will contain all the protein sequence information of mouse. Whereas in mouse genome GTF file, so gene text format, so it will contain all the annotations related to the entire genome of the mouse, and then there is a contaminant list. So this is just done to, this will be used in the later step just to make sure that the customized data set is properly generated, it is not from the contaminant. So now I have my gene text format as well as my proteome data. So I will, in terms of HiSat2, I require a genome, reference genome, so that will be given by the GTF file. So now assembly will be done between the FASTQ files, that is already available RNA-seq files, as well as the mouse genome file. So mouse genome will be kept as a reference, and on it other sequences will be performed, so that my sequences could be assembled to a single bigger sequence. So that will be done by HiSet2. If we say string tie, so string tie is a tool which assembles RNA sequence data into larger transcript sequences and quantifies their abundance. So similar to HiSet2 only, string tie, it joins those RNA sequences and it constructs a larger transcripts. So in HiSet2, I am bringing my RNA sequences, I am placing them on the genomes. I am doing assembling at this level of HiSet2, but in case of string tie, I am kind of identifying that from which transcript they are coming. So that is done by the string tie. Now I have performed my assembly and analysis. So in terms of free base, it will look for the variants. For example, in assembly also there are some sequences which might not set well. There are some changes at some positions. For example, some nucleotides might not match. So in that case, they can be a variant. So in that case, I cannot ignore my entire sequence if single nucleotide is only variable among both of them. So in such scenarios, freebase is used, which will look for the variants, that in which positions the nucleotides are varying. So it will capture the variant information. It will also keep track of the information. So now I will generate custom pro database, so customized protein database. So after joining them, means HiSet2 is assembling my smaller RNA sequences into larger units. So now after assembly has been done, I will generate the customized protein sequences, because this is the data taken from the RNA-seq study. So each sequence will form an entire RNA sequence. So that entire RNA sequence can now form a protein sequence. So now if RNA sequence is obtained, so its custom ProDB will generate what could be the protein sequence that could be generated from that RNA. So custom protein sequence would be generated here. If we talk about string tie, so after assembling, if a larger transcript is formed, so string tie will try to identify that which transcript it is coming from. So it will compare it with the GFF. So GFF is also another file which is gene format file, which annotates the genome file only. So in GTF, it tells you the coordinates, that where exons are lying, whereas in GFF, along with the coordinates, it also tells you which is the exon position, which is the transcript. So string tie will compare all of that longer transcript sequence, and it will identify that what is the transcript which is present in the already available data set, and it will also translate that transcript. So it will convert that transcript into a protein sequence. Now similarly, both of them will feed their data into a custom protein FASTA database. So now I have customized my protein sequences. They are generated from my RNA-seq data, and these are customized. These are not the data coming from the proteome sequence database. They are generated from the RNA-seq database. In case of string tie, they are compared to the already available annotations. So in that case, I will not find the non-canonical sequences, whereas in terms of HiSet, because I am not comparing it that whether my sequence is correct or not, so in that case novel protein sequences can also be generated. So now my custom protein sequence database has been generated. I am also using contaminant list, because in terms of mass spectrometry data analysis, we require some kind of decoy sequences to make sure that my peptides are aligning to the correct. They are not randomly getting aligned to a sequence, they are confidently aligning to some sequence. For that we require the contaminant list. Yeah, there are some other things, which is custom ProDB. So it is a R based tool that creates custom protein databases by incorporating genomic variants and RNA-seq data. So this is also a tool actually which converts, so as we have discussed, that it converts the RNA-seq into a protein sequence. Then GFF compare, which we have seen in this case. So GFF compare is also a tool which compares the RNA-seq transcript assembly to a reference annotations, which provides novel transcripts only from the assembly data set. So it means that in my assembly, if that particular sequence is present, then only it will give me the transcripts, otherwise it will not give me any transcripts. So new transcripts won't be generated. It can only give me transcripts which are already available in the assembly data set, and assembly data set is the one which is obtained after assembling to the already available GTF, uh, already available reference assembly of the genome. So is it clear, or should I again repeat it, because there are many things which are present in this particular step. Okay, so now let's look into the next step, which is the sectioning workflow to reduce the RNA-seq FASTA database size. Yeah, so now I have generated my custom protein FASTA database. So now there might be multiple transcripts which are of no use or which might not be accurate. So I will reduce the size of this particular. So this comes under kind of a data pre-processing step, in which I am trying to pre-process the already available data. I am trying to transform my data or filter my data. So in this case there could be n sections, n multiple sequences which are available. So I am taking one by one. This is a row MSMS data. Now search GUI and peptide shaker are used at this step. So what is search GUI? Search GUI is a graphical user interface tool which facilitates the identification of peptide spectrum matches by integrating multiple search engines for mass spectrometry data analysis. So now this row MSMS data is coming from my previous means in the first step where I have shown you, in the first figure where I have shown you that how they are collecting the data of, they are also obtaining the protein sequences. So after performing trypsin digestion, they have performed LC-MS analysis. So now the data which is coming from LC-MS analysis will be my row MSMS data, which is coming from experimental studies. So this is not a customized protein sequence, but these are the peptide sequences which are generated by mass spectrometry. So now my mass spectrometry peptides are also coming, and my customized FASTA protein sequences are also coming. So search GUI will try to identify that these peptides are belonging to which of the protein sequence. So search GUI is like identifying the peptide spectrum matches, because peptides are obtained in terms of spectra. So I have to convert that spectra or mass to charge ratios into a sequence. So that sequence conversion is done by search GUI. It tries to identify the peptide spectrum matches. Then second tool is peptide shaker. So peptide shaker will take the output from search GUI, which will be a peptide sequence. It will perform downstream analysis like false discovery rate estimation, protein grouping, and result visualizations. So downstream analysis is done by peptide shaker. So now after giving me the sequence of the peptides, this next step is the pass matching criteria. So in this there will be some matching criteria which will be defined. For example, these many sequences should be present, or depending on the what is the mass to charge ratio of the entire bigger sequence, and now after sequencing what is the mass to charge ratio adding up to. So there will be some matching criteria which it has to fulfill. So if it fulfill that criteria, so there will be some X matches. So X matches means from this particular N section, which will contain multiple customized protein sequences, my row MS data can map to some of those sequences. So in that case, number of sequences to which it matches will give me X number of matches, or X number of customized protein sequences from where it is matching. Now I will concatenate peptides. So concatenation means I will join those peptides, maybe to generate a bigger sequence, bigger protein sequence. Now in the next step, if for example if my matching criteria is not passed, so in that case I can discard it, or I can generate X random non-matching peptides. So non-matching peptides could be generated which are not matching. Then those peptides will also be concatenated, because concatenation is nothing but it simply means you have multiple overlapping peptides. So you will construct a single sequence which will come from multiple peptide spectra which are you observing. So in that case you are concatenating it to a single sequence, single bigger sequence. So now complete n times, you will repeat it n number of times, so you will get multiple sequences. Now from Swiss Prot mouse proteome, so Swiss Prot is like a database which already contains the protein sequence information which is already available. So it is like a reference data set which is available, and the data set which is coming from the peptide spectra and after matching it to custom protein FASTA database will be a customized database. So that is not already available. It might have certain known canonical peptides in it. So we will merge both of them. Now it will construct a sectioned custom FASTA database. Yeah, so now the next step is, did somebody, okay, GUI and PS are separate tools, can use separately. Yeah, they are separate tools, but what I can say is that the peptide shaker, it requires the sequence of the peptide, so it requires the peptide sequence, which can only be provided by GUI. So in cases where you already have peptide sequence, then you can use peptide shaker directly to perform downstream analysis, but these are mostly like interdependent on one another because they require the output of one of them. So GUI's output is required by PS, but GUI is independent of peptide shaker. Yeah, so as we have also seen in terms of freebase and HiSet, no? So freebase and HiSet are like, freebase is just used to look for the variants. So similarly, they are, means freebase is taking the input from HiSet and then comparing for the variants. Similarly, in terms of my search GUI also, it is giving the peptide sequence, and peptide shaker is trying to look that whether there are false positives or false discovery, what is the false discovery rate. So it is performing downstream analysis, it is refining my peptide spectrum matches. So they are separate. Now identification and verification of known canonical variant peptides. So I have generated my sectioned custom FASTA database. I have given it to search GUI and peptide shaker. RUMS data was also giving me the mass spectras, which will be converted to the peptide spectral matches or peptide sequences. Sectioned custom FASTA database is also taken. Now peptide spectral matches will be generated. They will be passed to PSMs in conventional proteome. So this is a bigger data flow. There are multiple data flows here. So this is a bigger so third stage or third workflow, which is telling me what is done here. So peptide spectral matches will be obtained. Now it will check that whether my PSM's peptide spectral matches are present in conventional proteome. So conventional proteome means already known protein sequences which are coming from SwissProt. So SwissProt contains already available protein sequence information. So after peptide spectral matches, because I have concatenated my peptide sequence in my previous step, means I have combined them, I have generated a single transcript or single protein sequence which is coming from the transcript level information. So in this case also I will have a protein sequence, and in SwissProt also I will have a protein sequence. I will compare them and I will identify that whether PSMs, the spectral matches, whether they are coming from conventional proteome, because my aim was to identify non-canonical peptide sequences. So if my PSMs in conventional proteome, then I will discard them because my aim is not to identify the already available protein sequence. Whereas if it is not in conventional proteome, then it will be a known canonical peptide spectral match. I will perform BLAST P. So as we have seen in our initial weeks, that BLAST P is a protein blast. So it will blast against all the available protein sequence information. So mouse genome matches, it will match to the mouse genome. If 100% match to mouse proteome by BLAST, so for example, if it matched to any of the protein sequence present in the in the BLAST database, if there is any protein sequence which is present in my PSM is matching to that, so in that case also I will discard it because I want to make sure that it should not be present already, it should be known canonical. If it is not, then it will be a known canonical PSM. I will use PEP query. So PEP query is also a tool which is used for the targeted validation of peptide spectrum matches from mass spectrometry data against custom peptide databases. So similarly, this PEP query will contain the PEP query will match my known canonical PSM with already available peptide sequences, so that it can make sure that this particular peptide sequence is not present in any other peptide repositories or datasets. So pass matching criteria. If it passes, then it is a validated known canonical PSM. If it does not matches, we will discard it. So in this scenario, this PEP query is the targeted validation of peptide spectrum matches from mass spectrometry data against the custom peptide databases. So maybe in this step they are trying to make sure that this particular peptide should already be present in the custom peptide databases so that it could be validated. So in this scenario it is not ignoring it, that it is making sure that it should available in that custom peptide databases so that the peptide is a valid peptide, it is not non-valid. So that is done by the PEP query. So ultimately the output will be a validated non-canonical peptide spectrum matches. So this was their entire pipeline, which I have divided into 3 stages, and I've kept in 3 different slides so that it could be easier for understanding. Yeah, so before moving forward I will again ask that whether it is clear till here. Okay. Yeah, so in this way it defines that how pipeline is different from a workflow. Workflow is like it could be manual also, it is also sequential stepwise thing, but in pipeline it is entirely automated and it could include multiple stages or multiple smaller workflows which are integrated to one another, which are joined to one another. So this was about the paper. Then there is another term which is a data model. So these things are quite confusing when we see workflow, pipeline, data model. These appear kind of a similar things, but they are different from one another. So what is data model? It is a conceptual framework that defines how data is structured, stored, organized and accessed within a database or information system. So it is kind of a, it is telling you the structure, what are the main elements and how they are related to one another or how they are accessed. So it is like a blueprint which tells you how data elements are related to each other and how they are processed and managed. So this is a example in which I have shown that what exactly is data model. So there are some alleles, if their observable phenotypes are recorded, so it will construct another data set which will be a molecular and cellular phenotype data set. Then there is clinical phenotype, which will be integrated functional measures coming from molecular and cellular phenotype, and they are also linked with alleles. So this is kind of multiple data elements, they are linked to one another, and these things are mentioning that how they are related, what is the points which relate those data elements. Then there will be information of individuals, so which will be a physiology. Individuals means after observing the physiology of individual, its clinical phenotype can be observed. If I look into the genetic makeup of the individual, so it will tell me about the alleles which are present in the individual. Similarly, environment, so these are the non-genetic factors which affects the individuals. So these boxes are representing my different bigger data elements and how they are linked to one another. So these arrows are representing their relationship to one another, and these things which are written, they mention that what is the relationship. So treatment protocols, so individuals with treatment protocols will be linked to drugs, that what are the drugs given to these individuals in which clinical phenotypes. So this all data is linked to one another, and these are the elements of the data. So yeah, so this is a data model which is not telling you sequentially flow of information, but it is telling you relationship of different data elements and what are the different data which are present and how they are organized or stored. And this is a kind of a structure of that. Now here is a comparison table between workflow, pipeline and data model. So this is a simple definition which we have seen, that workflow is like series of steps for processing proteogenomic data. So in this case, if I am processing proteogenomic data, in terms of pipeline, it is an automated sequence of processing steps, and data model is like structured representation of data elements. If you look at the focus, that where they are focusing, so workflow is focused on managing and automating the steps for data acquisition, analysis and integration. Pipeline is automating end-to-end process from raw data to analyzed results. So it is automated end-to-end. There will be no decisions which will be manually taken in between or anything. It will be completely automated. Data model is structuring and defining relationships, which could be between genomic, transcriptomic and proteomic data elements. So that is the work of data model. Purpose is like streamline and standardize the complex data processing tasks. Similarly, pipeline is also efficiently process large-scale proteogenomic data through sequential steps. In data model also, it ensures consistency, integrity and accuracy, that how data is stored and related. If we talk about components, so in workflow the components are the steps, which could be quality control, normalization, variant calling, decision points, manual inputs, automation. In pipeline, the stages are data ingestion, peptide identification, variant annotations, transformations and outputs. So these examples are given in terms of proteogenomic data analysis, so they could vary. And in data model there could be entities, attributes. So entities are like genes, proteins, peptides, and attributes are the properties of these entities. For example, genes can have gene IDs, proteins can have protein expression, protein IDs, or different things could be associated with the proteins. Similarly with peptides also, and the relationships, gene protein interactions. So these are the components of data model. And here is the example. So workflow for integrating the RNA-seq data with mass spectrometry data for peptide identification. So if we integrate them and we perform it stepwise, so that is a workflow example. Whereas in terms of pipeline for variant calling in genomic data, followed by the peptide quantification and differential expression analysis. So example of pipeline we have already seen. Whereas in data model, that defines how peptides are linked to genes and transcripts in a database. So that could be an example of data model. And if we look at the tools and environment, so which are the tools which we can use? So in terms of workflow, we can use tools like Galaxy or K9 to create and manage workflows that combine various analytical steps and softwares. In case of pipeline, we can use tools like Nextflow, SnakeMake or Apache Airflow to automate and scale data processing tasks. In terms of data model, we can use the relational databases like SQL or graph databases, which can generate a graph, which can generate a relationship between data elements. So these are different tools used to generate the data models. Now we will look into the big data analysis. So in this session we will be talking about big data analysis, and I think it is mostly covered in the week 7. So in week 6 also it was little bit talked about, so I have kept it. So big data analysis means when you have a bigger data which cannot normally come in your, for example, if you are running something on Excel sheet, the comparison, so if your data is in millions, so in that case it cannot come in your Excel sheet, it will not, you will not be able to open it or access it. So in such scenarios it is known as big data, which is difficult to handle in a smaller machines or smaller processors. So in that case we need some bigger machines, or it is a complex data set, so which is referred to as a big data. And big data analysis means when I am processing or examining that larger or complex data, which could either be done to uncover hidden patterns or identify correlations, trends. For example, if I am looking for all the people across the globe and I am trying to identify cancer genes and what are the variants, so that will be a huge data. So such scenarios, in that scenario I will perform big data analysis, and it can help me to identify the hidden patterns or identify correlations, that whether those genes are correlated or whether how epigenetic factors are playing roles. So all that comes under big data analysis, which can help us to means identify or generate insights which can drive decision making and strategic actions. Or it can also help us in terms of disease, it can also help us to generate personalized medicines. So that is also comes under big data analysis, and this involves using the advanced analytical techniques such as machine learning, data mining, predictive analysis, predictive analytics, and statistical analysis. So these all are different techniques which are used when I am analyzing the big data. So here is the big data characteristics. So when we have a large set of structured, unstructured or semi-structured data which is continuously generated at high speed and in high volumes. So in the lectures also, that person has also mentioned that, for example, I think Ratan sir has mentioned that in Google when you search multiple things, so you are simply searching it, but it is stored somewhere, that which is what is mostly searched by this particular person. So each and every click of yours is stored somewhere. So in that way there are multiple things which are people, there are multiple videos getting uploaded, multiple movies coming up, so that is adding up to the data. so which is adding up in high speed and in high volume. So that is also contributing to big data. So what are the different characteristics which defines that your data is big? So first is volume, which means what is the size of the generated data, what is the volume, what is the size. For example, one particular file is 20 GB or 10 TB, so that is the volume. When you are talking about the size, means what will be the storage space it will occupy in your system. Then velocity. Velocity means speed at which the data is generated or processed. So velocity will tell you speed, that how quickly data is being added. For example, if I am looking into the CP tag, so in that way, how many, means how the data is getting added to it, how many new genes, new tissues information is coming up each time. So that is known as velocity, with which speed new data is coming. Then variety. Variety means how diverse is that data which is available, it is structured, it is unstructured, how it differ from one another. So that is variety. And veracity means that how truthful or accurate and reliable that data is. For example, multiple people are uploading their peptide spectra, for example. So we also need to see that how reliable is that. Maybe somebody has entered some spectra matches which might not be right, or some data which might not be accurate. So in that case, veracity will also measure that how truthful or accurate the data is, because in big data there is no restriction, anybody can uh, contribute anything, anybody can add anything. So in that way, veracity will also make sure that it will check that how truthful is your data is. So these are 4 V's which are involved in big data characteristics. So this is a small representation that what is big data analytics, because you have a huge amount of data, and your ultimate aim is to like make it a useful smaller place, a useful smaller conclusion you want to make it from a bigger data. So big data could be in the public data, which is in the public repositories, or all the data which is available for the public for free. Or it could be a sensor data, it could be a structured data. Structured data is the one which is organized properly, which where you can look that this particular field has these values, so it is a structure, it is organized. Whereas there could be some unstructured data, randomly some values are present, numbers are present, you do not know anything, so that is unstructured data. And there are multiple, means there could be multiple sources from where this big data is coming. Then comes data analytics funnel. So data analytics funnel will try to check the big data. It is like a funnel, it will only pass the most appropriate data, it will remove the other data set. So it will decrease the volume of the content and increase the quality of the content. So in veracity, as we have discussed, that in big data any amount of data could be present, but how much data is relevant, that is checked by veracity. So in this case also, from the big data only relevant data will be kept, other data will be skipped, it will not be considered. Okay, yeah. So we will decrease the volume of the content and increase the quality of the content. Structuring and linking will be done. For example, in PCA also, which we discussed in the previous session, so what can happen is you have multiple genes and their expression information available. So in PCA also you try to identify the plane or directionality where the maximum change is observed, so that you can reduce the dimensions, you can reduce the number of features which you are considering. So similarly in big data also there are multiple features which might not be relevant, so you are trying to skip them, you are only keeping the things which are related, which are relevant. So you will increase the quality of the content, synthesis and enhancement is done. So what are the benefits of this big data analytics? Cost reduction, faster better decisions, and new products and services. So this is just an example uh, of the benefits if we look in terms of services and goods. But in terms of our data set, the benefits will be we can precisely capture the smaller set, which will be more accurate or more relevant set. We can generate personalized medicines, and we do not have to focus on all the proteins, we will simply focus on the key driver proteins or candidate proteins which are involved in particular cancer or all those things. So that is the advantage of big data analytics, which is taking the bigger data set and giving you the refined information in the end after performing the analysis of the data. And it will be better. For example, if my data set is small, then I may not be able to interpret in a better way. But if my data set is huge, so that means if I will compare it, I will make some decision by looking at that huge amount of data, so my decision will have more value. So that's why big data analytics is important, and that's why it is being studied nowadays. That's why the collaborations are made, that from different repositories data is collected in a single data portal, in a single common repository, so that anybody can access from the different parts of the world, depending on their need, on their desire, that which information they want to fetch or they want to process, they want to keep. So this is all about the big data analytics. And there was a paper published in Proteomics which was talking about this entire big data analysis, in which they have used the metabolomics and proteomics dataset. So this paper is the scalable data analysis in proteomics and metabolomics using biocontainers and workflow engines. So in the later slides we will also look into the biocontainers. So biocontainers are like, for example, in real world we see containers, they can hold multiple things. Similarly, in biocontainers, this is also like a container which can hold multiple softwares, multiple resources, multiple different version of apps. So that is the biocontainers. And workflow engines are like the workflows, the sequential steps which are being used. So in this particular review they have talked about the recent advances which have been made in the mass spectrometry and analytical methods, which involves integration of proteomics with big data science. That means from proteomic studies the data which is generated is huge, so it is contributing to big data, these mass spectrometry studies. So in this paper, in this particular review, their key tools are key tools for the metabolomic and proteomic data processing, and proposes using software containers. So in this also they have used the Galaxy and Nextflow to enable the scalable and reproducible large scale analysis, and their aim was to enhance data analysis in the proteomics and metabolomic communities. So in terms of metabolomics, this is a smaller workflow. So feature detection, library search, score filtering, quant metabolites, and downstream analysis. This is a smaller workflow. So metabolomics will have all the information related to metabolites which are coming from the studies. They will detect some features, search the library. So in the metabolomics, features will be searched, that what is the mass to charge ratio, what is the abundance. So these all comes under feature. Library search means already available metabolite sequence information is there, so it will, all those features which are detected at this step, they will be compared with the library which is already available. Score filtering will be done, means those matches, score filtering will be done to quantify the metabolites, that how many features were identified and matching to the library. So in that way, quant metabolites, this is a step which will quantify how much amount or what is the abundance of metabolites present in my sample. And then later on, downstream analysis. Downstream analysis means all those steps, including the false discovery rate checking, normalization, all those things related to analysis, it comes under downstream analysis. In proteomics, feature detection, database creation, database search, applying FDR, second database search, applying FDR, protein inference. So these all are kind of sequential steps which are performed with the proteomics data. So in this case, they are only mentioning you these steps, but what their main giveaway message is that metabolomics and proteomics, there is a big data. This is not a smaller data. So feature detection, so there will be multiple feature detection files will be generated. So this is a kind of big data analysis pipeline or big data analysis workflow. So if I will automate it, then it will become a pipeline, and if there are some manual steps involved, then it will be a workflow. So in this particular paper only, I guess they have mentioned that which tools they have used. So MS based tools and modules. So first tool is feature detection, RT alignment, database search, FDR filtering, protein inference, and then there are containers. So there is a package known as Bioconda package. For example, in your lectures also, it was being discussed that, for example, if one person is using some program, it requires some version of your R package or Python programming, it requires particular version, and in your device the version is, for example, I require third version, but my device have fourth version. So I again and again have to install particular version depending on which app I am using currently or which software I am using currently. So in such scenarios, it can disturb my entire system, because I can work with multiple files. So in each and every case, whenever I'm using some software, I have to change my version. So to avoid that, I can use Bioconda. It is like a small package which already contains all the required libraries or all the apps which are required as a background apps to to run a particular software. So Bioconda package is like a, it will create a small environment in which I can keep my versions of particular apps or files which are required for the means functioning of that particular software with which I am currently working, so that I do not have to again and again delete and reinstall apps according to the versions which are required. So this is like a container, a smaller environment. So there could be multiple environment on a single device, and in each environment you can download different versions of same app, so that your entire machine is not disturbed. So for that, Bioconda package is used. Similarly, in bio containers also, the Docker container. Okay. Yeah, so Bioconda is like, it comes under Anaconda only. So in Anaconda, as we generate environment, similarly it is called Bioconda when it is means handling the packages related to biology. So that's Bioconda. So it will contain all the packages or the libraries. For example, in Python also we have multiple libraries, so it will have all those libraries which are especially related to the biological analysis. So as we have BioPython, so that means the BioPython will be more dedicated towards biological data analysis. So similarly, Bioconda is under the Anaconda only, and it will involve more and more libraries related to biological analysis. So in Bioconda package, we will have different bio containers. So then there is a Docker container. So in Docker container also, multiple different apps or different files, different repositories which are required for the functioning of a particular software will be present. So in Docker container, multiple tools, multiple libraries, they will already be present, so that with a single click I can install them at a single step. I do not have to one by one select a particular library, then download it. So Docker container is like that, and they all are merged into a singularity container. So it comes under bio container only. So in their analysis, they have used Bioconda and bio containers to analyze the data, and these are the executors: Amazon Web Services, OpenStack, Kubernetes, cloud infrastructures, LSF, IBM. So in their analysis, they have used these executors, and local and single servers were used. So I have a peptide spectra matches which are I have a after mass spectrometry we will have a profile in which the peptide spectrum will be present. So feature detection will detect the features. It will like a search UI, it will identify that what are the features, what are the mass to charge ratios, and it will map it with the already available library, so that peptides could be identified. It will be searched against the database, and standard file format will be mzident.ml, so which is like mass to charge identification. ML is like machine learning approach, which will identify, which will compare mass to charge ratios and already available database sequence, that what should be the sequence of the peptide spectral matches. So peptide identification will be done. In this case, feature maps are generated, all these are combined, all these information are combined, and ultimately inference will be made for that particular protein. So what we mean by inference, for example, there might be some post translational modifications or all such things. So they can come from feature maps and after matching the peptides. So all those things, all those analyses are done in this way, and ultimately it will be inferred that what protein is present in which, what will be its abundance. So in that mzquant.ml, so that is also another tool which is used for quantification. So in this they have not mentioned it, but there are multiple tools with which different level of analysis can be done with my mass spectrometry data. So this was about their paper, that these things they have done to perform big data analysis. And they have also given with the table that means collection of public Nextflow and Galaxy workflows for proteomics and metabolomic data analysis. So as I mentioned, like Galaxy will also provide you different workflows, and you can analyze it by following the stepwise information which is given in it. So in Nextflow and Galaxy there are multiple workflows which are available. For example, if you have the data related to metabolites, so you can use their workflow metabolomics, which will process, quantify, annotate, identify, and perform statistical analysis of your data. And main tools which are involved in that particular workflow are XCMS, OpenMS, Camera, Medfrag. So these are some of the name of the tools which are present. And if you want to see it, so you can click on this link and you will be redirected to that Galaxy workflow, and you can see that how metabolomic analysis can be done using the Galaxy workflow. Then MZ quality, if you want to look for the quality of the mass spectra MS data which is generated, and the main tool is MS Vet FC. This is the link. Similarly, eco metabolites, so MS metabolites for ecology applications, ecomet is the tool. Fluxomics, stationary, 13C, MS, isoflux. So these are some of the example of workflow depending on that which analysis you want to perform. Similarly, in the proteogenomics, the description is proteogenomic analysis, and tools which are used in the proteogenomic analysis are the custom ProDB, peptide shaker, search GUI, MS convert, MSGF plus. So we have already seen that how proteogenomic analysis pipeline we have seen. So in that we have also come across some of the tools. So these are few tools which are used in Galaxy and Nextprot. So this is in the Galaxy. Similarly, metaproteomic analysis, metaquantome, unipept, metaproteome analyzer. Similarly, so in this table you can see that which are different workflows provided by Nextflow and Galaxy, which are especially related to the mass spectra data analysis as well as proteogenomic data analysis. So by clicking on these links, if you are interested, you can go through them. They will simply tell you the workflow, that how are these steps being performed. Now we will talk about firecloud. So before moving forward, I'll again ask that is everything clear till now? Okay. So now we'll talk about the firecloud. So as you know, this was developed by Broad Institute. So it is a cloud-based platform which is designed to support the management and analysis of large-scale biomedical data. So if we talk about Google Cloud, AWS, Microsoft Azure, so they are not specific to one particular data set. You can store any kind of information, you can perform any kind of processing there, but firecloud, it is especially dedicated to large-scale biomedical data analysis, and it is developed by Broad Institute. So it provides a scalable and secure environment for researchers to perform genomic and other data analysis in the cloud. So one advantage of cloud is that you can share your data to any person sitting anywhere in the world. Second thing is that you're not limited by the processor speed or space in your system. You can simply get any amount of space which you need, and on the basis of your requirement you can pay for it and you can access that space. For example, if normally I want to perform my NGS sequence data analysis, so I require a huge processor which could have multiple, its RAM should be of high, space, means it should be of higher storage space. So in that case I have to buy the machines which will be very costly, and I will not always be requiring that much space. So in that case, if I am buying it personally, so it will be very costly, whereas in firecloud or other cloud computing services, I do not have to buy them. I can simply rent them, perform my analysis. I only have to pay for little amount of time for which I am going to do that analysis. So it will be convenient for me and it will be cheap for me comparatively, rather than buying it personally. So that is advantage of these cloud computing, and this is the home page of firecloud. In this you can view their workspaces, you can view examples, you can browse the data which is already available in the firecloud, and it depends that there are some data sets or some workflows which are available for the public, anybody can access them. So depending on the criteria, that means how much data they can share, you can browse the data. So in this case, if we click on workspaces, so it will tell you all the workspaces. So in this case I have clicked on the featured workspaces which are present. So there are multiple workspaces which are already available. So Terra on GCP quick start, then there was COVID-19, so it appeared quite interesting as compared to others, so I have selected it. So this is a workspace which contains COVID-19 genomic data and workflows which are performing means analysis on this COVID-19 genomic data, and it was modified on 27th August 2024, and access level is reader. That means access level can also vary. For example, I am collaborating with some lab which is present in America, so in that case I can provide them access level to I can provide them access to change or to modify my data. So in that case access level can change, but now they have only provided reader level access, so that I can read their data but I cannot change anything. So in this way it is secure place. And firecloud comes under Google, means it depends that from where they have uploaded. So it is like Google Cloud, and Microsoft Azure can also be used as the background cloud engine, means where the data is handled. So if we click on COVID-19, then it will take us to the entire dashboard of that COVID-19, so which will tell you about the workspace, that what is the journal information of this particular workspace. So this will tell you that what information it keeps. So COVID-19 genomic data and workflows will enable you to perform viral genomic analysis on both the public data and your own data. So the advantage of these workspaces is that they already have designed some workflow that how to perform this analysis. So I can simply perform analysis on their already available data, or I can also take help of their workflow and give my data to that and see that what are the results, or I can compare, or I can combinedly give their data also and my data also and then check. So that this is one of the advantage of these workflow and workspaces, that I do not have to design my own workflow. I can already means take their workflow and use it on my dataset, and it will be quite quick and reliable also because it is already available. So if they have published their results, so that means their workflow is reliable, so I do not have to worry that the result of this workflow might not be right. So that's why. And then in the right side you can find the workspace information, that when it was last updated, when it was created. So in certain scenarios what can happen is that, as we know that in bioinformatics, means every time something new update is coming up, so new advancement is also done in terms of technology also, it is advancing day by day. So my workspace should be such which is regularly updated, means it should not be that much old, because if it will be updated, that means I can better rely on it. It might be using some new techniques or technologies, so in that case it is more reliable. So we can also check that when it was last updated, what was the creation date, and it provides you the information of access level. It also tells the information about cloud, that where in which cloud this information is stored. So in this case it is stored in the Google Cloud. It tells the location from where this information was submitted. So Google project ID, bucket name, bucket size. So there are few things which are given. It also gives information of owners, it also gives information of tags. So these tags are like keywords. For example, if you write it, so it number of tags can make your data more means visible to people. They can easily identify or search your data. So these are some of the tags. And in this they have given the workflow, that viral genomics workflow. So in this they have mentioned different steps, step by step, means how this analysis was performed. If we further click on the data, then it will also give us the data. For example, if I want to download their data and perform analysis, so I can simply click on the data and it will give me all the data. So these are the sample IDs, these are the accessions, aligned BAM. So BAN is like a binary alignment file, so binary alignment map. And this is nothing but, after sequencing, when you have multiple reads, so you try to align it to already available reference genome sequence. So BAM is also like the aligned sequences, which are after sequencing when you are aligning your reads. So BAM file will tell you that where your reads are aligned, so that a single sequence can be generated of the multiple reads. So that is aligned BAM file. Similarly, aligned BAM flag stat, so this will contain some statistical information, BAM IDs. So this is about their data, and I can download. It depends. For example, if somebody has huge amount of data and I just want to take a small sample, so I can select it. I can select these check boxes according to my need, or I can complete means select all also, and I can export their data. For example, if I only want to export their data, they already have done some analysis, so I do not want to run entire workflow. So in that case I can export the data, and in this they have given the sample. So there are 68 samples, and they have divided these samples into 4 sets. So there are 4 sample sets, and in the right side you can see the rate, so $0 per hour. So in this case you do not have to pay anything, it is for free, you can simply analyze it. But in certain cases you might have to pay. Now if we click on analysis, so it will tell us different steps or different how they have analyzed it. So mostly in this they have given the information of Python packages, because we have mentioned that in workflow it is like a script will decide that how the data is taken and how it is handled, how the processing is done. So in analysis it will provide you with all the codes. And why these codes are provided? Because in workflow it is not rigid. You at any point, if you want to change certain conditions, you can simply do it by making modification in the code, so that your means workflow is flexible. So for that they have provided the code, and by looking at analysis we can find the code. We can also download this code on our local machine and perform analysis, or we can perform analysis on their firecloud also. Now if we click on workflows, so there are multiple workflows which are used in this COVID-19 data analysis. So as we have seen in pipeline, there are multiple workflows which are linked, which are automated. So similarly there are different workflows. If I am interested only at one particular stage, so I can select that particular workflow and only run that workflow. But if I want to perform entire analysis, so I have to run these workflow one by one. So these are some of the workflows which they have used, which is present in their dataset. And yeah, so if I click on job history, so for example, how many people have accessed it, what is the download rate, so all that information can be fetched from here. And if I want to find a workflow, then I can click on plus, and I can identify some other workflow which is present somewhere else, or I can also use this particular workflow for my data analysis. So all those things can be done in the firecloud. So this was all about the firecloud. Now we will practice few MCQs. So before moving on MCQs, I will ask you that is there any doubts you have regarding this particular thing which we have covered, or any doubts related to assignment? Because rest everything which was present in your week, this particular week, was already covered in the lectures, and you have also done hands-on sessions as well. And about the targeted mass spectrometry and selected reaction monitoring, all those hands-on sessions have already been done, so I am not again repeating all those things. So okay, so is there any doubt, or should I proceed with the MCQs? Yeah, I have seen this question. So I have also searched for this, that positive control and negative control, so which temperature should be chosen. Even I could not also find the proper temperature, that what should be the temperature, because the temperature which they have given, it is quite closer to one another. But what I have found is that in terms of better differentiate between the positive control and negative control, temperature should not be very low and it should not be very high. So from those options you have to select one of the temperature, which I cannot give you the exact answer, but I can tell you that yeah, I have also confused ki it was not taught anywhere. Even I have also not come across any of such video lectures where it was mentioned, and not even in week content, but I have also searched it in the internet also, but I could not find answer. Yeah, you can ask them to, in the discussion forums only you can ask them, because I'm not the official TA, I am another TA, I'm not of that institute, but the official TA and other people, they can do something regarding this, because it was not provided. So yeah, they should take down this question. Yeah, but for guess you can select the temperature which is not extreme and which is not very low, so you can see accordingly. Okay, so now let's practice few MCQs. So skyline, actually I have not taken skyline into this session because I thought that it was already provided hands-on session by that guy from IIT Bombay. So have you seen those supplementary lectures in which they have, I think, talked about the skyline? Yeah, so that's why I have not included skyline into my today's session, because I thought that all those things are already discussed, so repeating them won't make any sense. Yeah, so yeah, so in the skyline you have to give your data and then it will acquire the data and then it will set up the methods. So data entry is done by you, rest acquisition and the methods you can select from the skyline software only. So it has different methods or different types of analysis which you can perform on the data which you provides. Yeah, so now let's look at this question, which is like, what are the advantages of scripted workflow in big data analysis? So first option is automated repeated tasks. Option B is it increases the human error. Option C is it increases auditability and reproducibility. And option D is it reduces the time taken for analysis. So you have to tell what are the advantages of scripted workflows. So option 1 is that A, B, C are true. Option 2 is A, B, D true. Okay, A, C, D. Yeah. So C, that means 3, right? Okay, so option 3 is true, which is A, C and D, because automated repeated tasks increase auditability, reproducibility and also reduce the time. It will not increase human error because it is scripted. So that's why option 3 was right. Now the next question is, what are the uses, advantages of cloud computing? So first advantage is data storage. Second option is big data analytics. C option is does not depend on internet connection. Option D is data backup and archiving. And option E is software testing and development. So you have to tell the advantages, and these are the options which carry the points. So you have to tell which one is the advantage. So what do you think will be the right option? Can you guess? Okay, now let's see. So the correct answer is, okay, so the D or these things, DU means points, right? 1, 2, 3, 4. So D means this, which you guessed. Okay. Yeah, so right answer is option second, which is A, B, D and E and not C, because in cloud computing it depends on internet connection, because cloud computing main thing is that multiple devices are connected over the internet connection, so it requires, it depends on internet connection, so this is not the advantage. Rest all are the advantages of cloud computing. Data storage, you can store the data. If we have big data, then we can analyze it. It provides data backup, archiving and software testing and development. Now let's see next question. So which of the following is incorrect in regards to firecloud? which is NCI cloud resource. Option A, applicable for biomedical research only. Option B, firecloud allows data accessibility and collaborations. Option C, firecloud don't allow any analysis and is restricted to data storage only. And option D, both A and B. So you have to tell which one is incorrect. And for creating confusion, they have kept option D. So what do you think will be incorrect? Because you can simply cancel out the options. We have already discussed firecloud. So any guesses? Option C. What about polemy? Do you want to make any guess? Okay, fine. Yeah, right. Yeah, so both of you have guessed right. So yeah, option C is the incorrect one, because it also allows us to perform analysis, as we have already seen the different workspaces. So um, it is allowing us to perform the analysis, and it is not just restricted to data storage. So this option is incorrect. And they have also kept option D, which is both A and B. So if you do not read the question properly, for example, if you skip the incorrect option and you simply look at firecloud and then take option D, so means for these things you should make sure that you read the question properly and do not get confused by the options. Now let's see next question. So which of the following file format contains gene description and its related features? So it's like gene description and its features, and we have also discussed it today. Okay, pasta file, MGF file, GTF file, and MGML file. So Polemy has guessed it earlier only. Okay, so what about Srijita? What do you think? Yeah, so right answer is GTF file. So it is a gene transfer format. It is a file format which is used to hold the information about gene structure. So when we say gene structure, it means it incorporates the UTR regions, exon, intron, all that information, promoter region information. So that all information is being stored in gene transfer format. It will simply contain the coordinate information, that from where to where this particular region is present, along with the chromosome level information. So it is a tab delimited text format based on the journal feature format. So yeah, so there is another file which is the journal feature format. So these both gene transfer format and feature format, they are kind of similar files, but means GFF file is more feature based and it is more reader friendly. That's why people mostly prefer GFF over GTF. And a significant feature of GTF is that it can be validated. Given a sequence and a GTF file, one can check that the format is correct or not, and it reduces problems with the interchange of data between different groups. So that is the advantage of GTF file. And this is the example of GTF format, that how it appears. So GTF file can be provided by different online available databases or data repositories. So in this case, Ensemble is a genomic data repository which is generated by the EMBL, and it contains the information of entire genome of multiple organisms. So in this case I am taking example of human. So in GTF file it will provide you information of start codon, from where it is starting. So these are the coordinate positions. So in chromosome, the position of nucleotide, and then it will tell you the frame, in which frame it is present. And similarly there are other multiple features which are telling you about gene IDs, its version, its transcript ID, transcript version. So versions are like every time when you perform the sequencing or genome level sequencing is done, so versions are updated every time whenever some new information is available. So whenever updates are made, so versions keep on changing. So ultimately GTF file will tell you the start codon, exon, CDS is the coding sequence, then exon its coding sequence. So this is all about the GTF file, gene transfer format, and what information it keeps. So now let's look into the another question. So which of the following statements is not true about a selected reaction monitoring experiment? So option A is SRM experiments can be performed only in Orbitrap instruments. Option B is the software Skyline can be used to set up the experiment and analyze the datasets. Option 3, collision energy optimization and transitions selections are linked with SRM experiments. And option D, SRM experiments is a popular approach of targeted proteomics and facilitates the validation of biomarkers. So which is not true about selected reaction monitoring? Because I think in the supplementary lectures you have seen the selected reaction monitoring, and you have also looked into the what is the selected reaction monitoring, and means it comes under targeted proteomics. Okay, so Srijita has guessed option A, which is okay. Both of you have guessed option A. So now let's see. Yeah, so the right answer is option A, which is not true about selected reaction monitoring, because they mentioned that SRM experiments can be performed only in Orbitrap instruments. So yeah, this is not true, and rest all other options are true. So now let's move to next question, which is Galaxy P is a multi-omics informatics platform which focuses on integrative analysis of genomics, transcriptomics and mass spectrometry based proteomics data. And here the figure is given which shows the proteomic data analysis using Galaxy software. So you have to identify what are these A, B, C and D options, and these are few of the options which are given. So from here you have to select that what should be A and what should be B. So as today also we have seen that in the pipeline we have seen multiple stages. So if you remember them, then you can easily find out that what should be A, B, C and D. So A should be protein FASTA reference proteins, and B should be somewhere related to peak list of MSMS data, and then they are integrated and coming to C, and D is like output which is getting generated. So take your time, look at these options, and what do you think will be A, B, C and D? So one hint I can give you, that here it's written peptide shaker. So you already know that what is the input for peptide shaker. So yeah, but I think this hint won't help you because in all of them C is already written as search UI. Okay, so Srijita has mentioned that D is right, and Polemy says C is right. Okay, now let's see what is right. So option D is right. So why? Because protein FASTA reference proteins. So as we have seen in that pipeline, that RNA-seq database, it will provide all the information of already available RNA sequences. So from the RNA sequences, we will generate custom protein sequences and peak list of MSMS data. So we know that the mass spectrometry data is stored in the MGF file. So in mascot as MGF files are used. So now both the files will be taken to C. C is like search UI, which will identify what will be the sequence of the peptides. So in this case we only have a spectra. Now I want to identify the sequence. So we have to compare it with already available database, so that particular reference proteins will be generated from the RNA-seq database by using custom ProDB. So it will generate customized protein sequences. Now these both will be searched using search UI, which will provide some sequence to the peptides. Now peptide shaker will perform downstream analysis. It will identify the peptides, and ultimately it will generate the peptides for blast P. So in this case, after performing blast P, we will look for the proteins where that particular peptide is present. So this is a integrative analysis, and option D is the correct. So now let's look at the next question. So this question is, what is a bucket in the context of cloud storage? So option A type of virtual machine, option B a container for storing objects and files, option C a database table for structured data, and option D a network security group. Because you have come across the bucket, so okay, option B. So Polemy, what do you want to guess for this? Okay, yeah, so option B is right. Bucket is a container for storing the objects and files. It is not a virtual machine or database table. So this is a representation, how to represent bucket, and bucket concept was mainly given by Amazon Web Services, and later on in some cases even Google Cloud also use it, but the main concept was given by AWS to represent the containers. So this is the origin bucket, and it can give information to destination bucket. For example, already hosted server is present in which there might be different objects or different files which are present, so we can simply use that bucket. So destination bucket is the place where I am taking information of original bucket. I can select few of the files which I want to take from the original bucket, or I can also select all the content which are present in the origin bucket, and that origin bucket can have certain permissions. So depending on the permissions which are available, for example, there might be some data objects or some files which you might not access. So in such scenarios you cannot access those files, or you cannot download those files, you cannot import them to your device. So ultimately the bucket is like a container which can store objects, files, or different softwares or different versions of different smaller libraries. So that is the purpose of bucket. So next question is, which of the following cloud storage services uses the term bucket to refer to a storage container? So option A Microsoft Azure Blob Storage, option B Google Cloud Storage, option C Amazon S3 Simple Storage Service, and option D is IBM Cloud Object Storage. Okay, so you have said option B, but right option is option C, which is Amazon S3, that is Simple Storage Service. So bucket to refer to storage containers. So in Amazon S3, when they say bucket, so they mention it to a storage container where something is stored. So mostly this concept was given by Amazon. Means in certain scenarios Google also use that, so you're not wrong. They also use bucket as a container where multiple files could be kept, but means Amazon S3 was the first one to give the term bucket for the storage containers. So when we say Amazon S3, it is a simple storage service in AWS which store files of different types, and these files could be anything. They could be photos, audios, or videos as objects, which provide more scalability and security. So instead of storing all those information on my single machine, I can simply keep it in cloud, so that I do not have to worry about space. They are secure, and any amount of data can be stored. It allows the users to store and retrieve any amount of data at any point in time from anywhere on the web, and it facilitates features such as extremely high availability security. So this is about the bucket. Now let's look at the next question. So what is Docker primarily used for? So you have also come across the term Docker. So option A is data analysis, option B application containerization, option C cloud storage, and option D network security. So what do you think this Docker is used for? Today I've kept a lot of practice MCQs because content was not that, so that's why. Okay, you've guessed option A, data analysis. What about Ptolemy? Today we have a lot of time, that's why I'm not rushing through the slides. So A, B. Okay, you are confused among A and B. Maybe you are thinking of both A and B. Okay, so now let's see. So the correct option is option B, which is application containerization. So I think you have seen this figure in the lectures when you are going through those lectures. So Docker is like a container, because in there only, in the Docker only, they have given the example that why we require Docker, and they have given the example that if I have to perform some analysis on my particular software, so they require some version of Python programming or some version of some library, so I again have to install it, delete it, install it, so that is difficult. So in that case, Docker was designed such that it can contain, it is also like a, as we have seen in bioconda environment, biocontainers. So similarly, it is also like a container where all the relevant softwares or relevant libraries are kept of certain version which are required for the functioning of apps. So instead of means you can simply create an environment and install the apps from the Docker and simply use it. So it is like a container. It simplifies the configuration, developer productivity, server consolidation, multi-tenancy, rapid deployment, debugging capabilities, app isolation, code pipeline management. So these are the advantages of the Docker, that what it does. But ultimately its main task is to hold all the applications onto one place, so you do not have to rush through other places, and you can simply install the Docker image and you are sorted. Okay, so the next question is, what is firecloud primarily designed for? So option A is web hosting, option B is data storage, option C is scientific data analysis and genomic research, and option D is machine learning model deployment. So these MCQs are quite easy, I guess, because we have already seen why firecloud was designed and what was the primary aim was of firecloud. So what do you think? Any guesses? Okay, option B. What about Ptolemy? B. So you both think that firecloud was primarily designed for data storage, but its correct answer is option C, which is scientific data analysis and genomic research. So their primary aim to develop or design firecloud was not to store the data, but it was to perform analysis, because it was made for the biomedical data analysis. So firstly they have designed it for the biomedical data analysis, and later on they have also integrated data storage and data sharing abilities to it. So primary aim was also the scientific data analysis. Data storage is also not wrong, but primarily it was not developed for storage, it was developed for data analysis, so that people sitting in multiple places can analyze the data. So right option is option C. Now question number 11. So which platform provides firecloud as a service? Amazon Web Services, Google Cloud Platform, Microsoft Azure, and IBM Cloud. Okay, Srijita has guessed option B. Okay, so waiting for Ptolemy. What do you think will be the right answer? Okay, you both have guessed option B. Yeah, so option B is right. So Google Cloud Platform provides firecloud as a service, and there is a name called Terra, so from where you can access the firecloud. So yeah, option B is right. So question number 12 is, in firecloud what does a pipeline refers to? there are lots of questions related to firecloud only. So option A network configuration for data transfer. Option B, a sequence of data processing steps or tools. Option C, a method for encrypting data. And option D, a tool for container orchestration. So what is pipeline? Yeah, it's quite easy. Yeah, you both have guessed it right. So pipeline is a sequence of data processing steps or tools, and in this it was quite easy to distinguish among all other options. But if workflow would also have been in the question, then it might become difficult. Okay, so question number 13. So what is WDL in the context of firecloud? So this was also covered in your lectures. So option A, web data language. Option B, workflow description language. Option C, web development language. And option D, workflow data logger. So what is WDL? So in firecloud they have mentioned it. Okay, yeah, so the right answer is workflow description language, and it is a language which is used to define and manage complex workflows, especially in the context of computational biology and data analysis. So it is a way to describe that how to execute a series of tasks such as data processing and analysis steps in a reproducible and automated manner. So in this figure I have shown that how the WDL language will represent. So this is a workflow input which could be reference genome, gene models, RNA reads, fruit fly forward and fruit fly reverse. So this is an example in which they have performed experiments on Drosophila, which is also known as fruit fly. So its RNA sequence reads have been taken towards forward and reverse, both side reads are taken, gene model information is available, and reference genome sequence is also available. So these are the inputs for workflow. Now in the processing, what are the processing which can be done? So trim low quality bases. So in this, the after sequencing reads are obtained, the low quality bases will be trimmed, they will be removed. So this is a filtering kind of a process. Now mapping will be done of these reads. So means comparing these reads to the already assembled or available annotations. So by using GTF and GFF files, the mapping will be done of RNA reads to the sequence, index alignments, quality control forward, quality control reverse. So all these things comes under processing, and ultimately workflow outputs will be BAM sorted indexed. So it will be the alignment file, quality report forward and quality report reverse. So this is a workflow description language which tells you that in workflow how the work is organized, how the tasks are organized. So it is a language to define and manage different tasks. Yeah, now so this was it. This was all the MCQs which were available. So if you have any further doubts, then you can ask. We still have 2-3 minutes. Okay, so I will look for this book and I will find a PDF, then I will provide that PDF. Maybe in the end of the slide, either I'll provide the link or I'll see that how can I share this. So don't worry, I'll check for it. Yeah, thank you so much. In each and every session, nobody is joining, only you two are there. I'm thankful to both of you that you are there. Okay, so that's all for today's session. So now I will end the session. Thank you so much, and I'll meet you in the next session. So by then I will search for this particular book. Okay, thank you.